Welcome back to this next video in which we are discussing valvular heart disease. Okay, so we're continuing on discussing pathological processes that can lead to valvular heart disease, and the next one in our list is infective endocarditis. So let me begin by giving you an overview of what infective endocarditis actually is and how it can lead to valvular heart disease. So, looking at the name here, endocarditis. That's going to mean inflammation of the endocardium. Now remember, the endocardium means not just the endothelium that lines the inside wall of the heart, but also that connective tissue underneath the endothelium before you reach the myocardium. So endocarditis is inflammation of that layer of the wall of the heart. Infective endocarditis tells us that the reason for this inflammation of the endocardium is going to be that we've got an infection. So infective endocarditis means you're going to get an infection of the endocardium, which is going to lead to inflammation. And as we'll see, the major causes of infective endocarditis are bacterial, namely Staphylococcus aureus and viridans streptococci, but we'll come on to that in a moment. So the other thing that I'd like to say right at the start here is that infective endocarditis is often abbreviated down to just IE. Right, so now let's discuss why can infective endocarditis lead to valvular heart disease. So remember, the endothelium that lines the inside of the wall of the heart is continuous with the endothelium that lines the surface of the valve cusp. So if I just draw a valve, a heart valve, so that I've got something to point at, let's have the mitral valve just as an example. So drawing the mitral valve from above again, so we're imagining we're in the left atrium and we're looking down at the mitral valve. Remember, the mitral valve is also called the bicuspid valve, and it consists of two cusps, the anterior one and the posterior one, and the posterior one is split into these three scallops, the anterolateral scallop, the intermediate scallop, and the posteromedial scallop. So here is our mitral valve. So the valve cusps are covered in an endothelium that is continuous with the endothelium that lines the wall of the heart. So if you're getting infection of the endocardium, that can also cause infection of the valves, basically, and it very often does, and this can lead to massive problems with the heart valves functioning. So infective endocarditis, the infection can spread to the heart valves. What can this lead to? Massive valvular destruction. So when you get these infections of the heart valve cusps, let's say the mitral valve here, you get the infection of the heart valve cusps, it leads to massive destruction of the cusps. So the cusps can shrink because huge amounts of them have been destroyed by the infection. So drawing that on here, imagine the cusps have now shrunk down to these purple lines here. And you can see maybe what this is going to lead to if that was the case. So the cusps can shrink down, so I'll just put this as one of the consequences. So cusp destruction output. You can also get holes forming in the cusps because the infection has rotted the whole way through the cusp. So let's draw a hole there. So you've got a hole in one of the valve cusps, and I'll draw another one of those on, so holes. The other thing that it can lead to, and this is only relevant, of course, if it's affecting the atrioventricular valves, is it can lead to the detachment of the chordae tendineae. So it can, the infection can destroy the connections between the chordae tendineae from the papillary muscles in the ventricles to the free edges of the valve cusps. So you can get chordal detachment, which of course makes the papillary muscles useless, because if they're not attached to the valve cusps, then it doesn't matter if they're contracting, they're not going to help to prevent the inversion of the valve cusps. So chordal detachment is another consequence of infective endocarditis spreading to the heart valves, and of course this is only relevant for the atrioventricular valves. The semilunar valves don't have chordae tendineae attaching to them, so that's not relevant there. Just the holes and the cusp destruction are relevant there. So, putting this all together then, if this was to happen to a heart valve, what do we imagine that is going to cause to the heart valve? Do we think it's going to cause stenosis, or do we think it's going to cause regurgitation? Clearly, it's going to cause problems with the heart valve shutting properly. Blood is going to be regurgitating the wrong way, because, as you can see, if they've shrunk down, then they're not going to be able to shut properly. There's now a great big gap here where blood can go back from, in this case, the left ventricle into the left atrium. Also, if we've got holes, that's not going to help. And if we've got caudal detachment, that's going to potentially lead to inversion of the heart valve cusps when the ventricle below contracts, either the left ventricle or the right ventricle, depending on whether we're talking about the right or left atrioventricular valve. And that, of course, will lead to blood going the wrong way as well. And so overall, all of these things here lead to valve regurgitation. So we've seen two pathological processes now that lead to stenosis, and we're now going on to a pathological process that leads to regurgitation instead. So just before we begin the discussion of the pathogenesis in detail then, let's discuss which valves are most commonly affected in infective endocarditis. Now, for reasons that I will tell you about as we go through the pathogenesis in more detail, it is again the left-sided heart valves that are affected more commonly than the right-sided heart valves, and I will explain why that is when we go through the pathogenesis. It's again because the left heart valves are under more strain than the right heart valves. We will later on see pathological processes that affect the right heart valves more than the left heart valves, such as carcinoid heart disease, which we'll do right at the end, but this is another one that majorly affects the left-sided heart valves. So which heart valve is most commonly affected? Well, you might have guessed from the fact that I chose to draw the mitral valve as the example that the mitral valve is the most commonly affected. So mitral regurgitation can be caused by infective endocarditis. After the mitral valve, it's then the aortic valve. So the aortic valve is the second most commonly affected, and you do see aortic regurgitation due to infective endocarditis. Then the left-sided heart valves, the right-sided heart valves, the pulmonary valve and the tricuspid valve, they're hardly ever affected, but they can in principle be affected. So I'll put at the bottom, it's the right side heart valves. Okay, so there's an overview of infective endocarditis and how it can actually lead to valvular heart disease and the nature of the valvular heart disease that it leads to, mitral and aortic regurgitation majorly. Right, let's now go through the pathogenesis in detail then. So, we need to begin by discussing the bacteria that can cause infective endocarditis. And here we need to discuss the fact that infective endocarditis is often split into two different forms. So, i.e. our right, for short for infective endocarditis, and there are two forms of infective endocarditis that we often distinguish, which are called acute and subacute. So acute is the more severe form, the more rapidly progressing form, and subacute is the more slow form. Now I want to stress that both of these forms of infective endocarditis, acute infective endocarditis and subacute infective endocarditis, they are both life-threatening. They are both very, very serious illnesses that will kill you if you are not treated. However, which one will kill you faster? It's the acute one rather than the subacute one. So what's the difference between acute and subacute infective endocarditis? Well, the difference is what bug is actually causing the infection. Okay, so in acute infective endocarditis, the bug that actually causes the infection is Staphylococcus aureus. And these, I'm going to go through acute and subacute infective endocarditis, and I'm going to say specific bugs that usually cause them. These bugs that I'm going to go over, these bacteria that I'm going over, these are the major ones that cause infective endocarditis. Very rarely you can have a form of infective endocarditis which is neither of the types of bacteria that I'm going to go through, but we're going to ignore those, we're going to brush those aside. The bacteria that I'm going to say here, which make this division of acute and subacute infective endocarditis, are the major causes of infective endocarditis. So, Staphylococcus aureus then is the major cause of acute infective endocarditis. So, as I did for Streptococcus pyogenes, let's just give a little bit of an introduction to Staphylococcus aureus. So again, it's a gram-positive bacterium. So, drawing it out here, it's going to have a cell membrane. It's again a coccus as well. So we see the coccus here, so that means it's a round cell. So it has a cell membrane, and then it will have a nice thick peptidoglycan cell wall around the outside, which I'm drawing in blue here. So it looks, as far as this picture is concerned, just like the picture I drew for Streptococcus pyogenes. Now, what does Staphylococcus mean? What's the difference between Staphylococcus and Streptococcus? Well, if you remember, Streptococcus meant that the bacterial cells grow in lines. It was telling us about the fact that if you look at a slide with lots of Streptococcus cells on it, they will grow in lines. You'll see these lines of cells. Staphylococcus do not grow in lines. They grow in these arrangements that look like bunches of grapes, and I believe it's Greek or some old language. I'm not a big one for languages, but in some old language, staphylos means grapes. So Staphylococcus means these round bacterial cells that grow in arrangements that look like bunches of grapes. So just drawing an example, it will look like this. You've got loads of bacterial cells growing and forming this bunch of grapes structure here, like so. Okay, so I think that gets the point across. So Staphylococcus aureus, when it divides, it does not divide in that beautiful arrangement like the Streptococcus cells, where you get these beautiful lines forming. Instead, you get this random way of division, which results in a bunch of grape-like appearance forming. Okay, so that's an introduction to Staphylococcus aureus. It's a commensal on the surface of the skin. Most of us are absolutely covered in Staphylococcus aureus, and it causes us absolutely no problem usually. Okay, so it's usually on the skin, so it's a skin commensal. It should not be anywhere near the heart, however, and in acute infective endocarditis it's going to get to the heart. We'll discuss how in just a moment, after we've discussed the bugs that are responsible for subacute infective endocarditis. So let me get another colour for subacute. So in subacute infective endocarditis, the major cause of the illness is bacteria known as viridans streptococci. Now, viridans streptococci are not a single species of bacterium. Staphylococcus aureus and Streptococcus pyogenes, they are a single species of bacterium. Viridans streptococci is a whole group of species, and an example of a specific species of bacterium that is in this group of viridans streptococci is Streptococcus mutans. So Streptococcus mutans is an example of a species included in this viridans streptococci. Streptococcus pyogenes is not included in viridans streptococci. Viridans streptococci, these are the major commensals in the mouth. So these are mouth commensals. And Streptococcus pyogenes is not really counted as a mouth commensal. It's present in very small amounts, but it's not a major presence. It shouldn't be a major presence. If it becomes a major presence, then you get strep throat. Viridans streptococci, they are major presence within the mouth. Okay, so they're all the species of streptococci bacteria that are commensals within the mouth. That's the definition of viridans streptococci. And then major example is Streptococcus mutans. You have a lot of this bacterial species in your mouth. Okay, but there are other species that are included in this group of species called viridans streptococci. So these are all streptococci, so the picture's exactly the same. They're all gram-positive cocci, and they will grow in the line arrangement because they're all streptococci rather than staphylococci. So these bacteria, this bacterial species and this group of bacterial species, these are the major causes of infective endocarditis. As I say, there are other bacteria, much more rarely, that can cause infective endocarditis. We're going to sweep that under the carpet. We're just going to focus on these two. Most cases of infective endocarditis are either caused by Staphylococcus aureus or viridans streptococci. If it's caused by Staphylococcus aureus, the disease is very fastly progressing. This is very, very virulent. It causes disease very quickly, and that's called acute infective endocarditis. If it's being caused by viridin streptococci, then the disease progresses less quickly. It's still very dangerous if not treated, but it's called subacute infective endocarditis. So if you were forced to have one of these, you would pick the subacute form of infective endocarditis. Right, so those are the bacteria that can actually cause this. We now need to discuss how do these bacteria, one that's a skin commensal and one that's a commensal in the mouth, actually get to the heart. So the whole process begins then with bacteremia. These bacteria have to get into the blood. That's how they're going to get to the heart. So we need to discuss how do they actually get into the blood. So either Staphylococcus aureus or viridin streptococci need to get into the blood to cause subacute or acute infective endocarditis, respectively. So the whole process begins then with bacteremia. And bacteremia means bacteria present within the blood. So these bacteria need to get into the blood, and that's how they're going to get to the heart. Now, I just want to clear up something that people often get confused about, which is the difference between bacteremia and septicemia. Bacteremia just means that you have bacteria present within the blood. Septicemia literally means blood poisoning, and this is much more dangerous. Bacteremia, the bacteria are just present in your blood. You've got a few bacteria maybe circulating in your bloodstream. Septicemia, you've got loads of bacteria present within your blood, and even worse, those bacteria are actually dividing within your blood. So the bacteria will be dividing within your blood, and you'll have absolutely loads of them. So your blood is poisoned, and septicemia is life-threatening. Bacteremia is not life-threatening. Just having a few bacteria floating around in your blood is not life-threatening, because they'll be cleared, hopefully, very quickly by the spleen or some other means. Right, what's going to happen then in infective endocarditis is we need to get bacteremia, because this is the way that these bacteria are going to get to the heart. So how do they actually get into the bloodstream then? So let's start with Staphylococcus aureus. So if you're going to get acute infective endocarditis, you're going to get a Staphylococcus aureus bacteremia. How are they going to get into the bloodstream? Well, the most common way that Staphylococcus aureus can get into the bloodstream is through using non-sterile needles, which is why acute infective endocarditis is quite common in intravenous drug users. So I'll again have two divisions here. So in the case of Staphylococcus aureus, the major way that it gets in is non-sterile, which means that there are bacteria present in the needle. So non-sterile needles. Okay, so if you don't use a sterile needle when you're injecting something into yourself, then that needle will almost certainly have Staphylococcus aureus on it, and then when you inject that into your vein, you will be putting Staphylococcus aureus into your bloodstream, and that's how you get bacteremia. And then that Staphylococcus aureus can go in the bloodstream to the heart. Okay, how do you get viridin streptococci into the bloodstream, which are going to cause subacute infective endocarditis? Well, they generally get in through dental procedures. You see, when you have a dental procedure done, you bleed a lot, and that means that the viridin streptococci, these commensals in the mouth, can actually get into the bloodstream through the open wounds. So dental procedures is the major cause of viridin streptococci bacteremia, and you do often see people getting subacute infective endocarditis after having some dental procedure done. Okay, so those are the ways then that these bacteria are going to get into the bloodstream. Now, of course, once they're in the bloodstream, they're going to go to the heart. They will circulate around and go into the heart. They'll first go to the right heart, so you might think, surely that's a reason that the right heart would be more affected. But then, of course, they're going to go through the pulmonary circulation and get to the left heart. So I'm going to give you the reason just now why it's more common for the left heart to be affected rather than the right heart. So, they've got into the heart chambers. how now are they going to actually affect the endocardium? How are they going to take hold there? Well, they cannot just bind to endothelial cells. Endothelial cells are absolutely fantastic at lining blood vessels and lining the surface of the heart. They do not allow bacteria just to stick to them. They don't allow much to just stick to them. They're incredibly good at just making a nice barrier there that nothing can stick to. So these bacteria cannot just stick to the endothelial cells and then start infecting the endocardium. So how then do they actually infect the endocardium? Well, they infect the endocardium at sites where there are pre-existing tiny vegetations. And these vegetations are going to be far more common on the left heart than on the right heart because of the increased hemodynamic strain on the wall of the left heart. So I need to now tell you, what do I mean by vegetation? So we now need to discuss this concept of a vegetation, and it's important that you've got pre-existing tiny vegetations. So we need a pre-existing vegetation. So what does this actually mean? So a vegetation is a place where the endothelium has been damaged to an extent where the endothelial cell has actually gone from that area. So let me try and draw a picture of this. So let me put some endothelial cells here. So here's one endothelial cell, and here's another endothelial cell. And you can see that there should be an endothelial cell in between them, but let's say because of hemodynamic strain on the endothelial cell, so because of blood pushing up against it continuously, one of the endothelial cells has been damaged and has gone from that area. So let's now put a basement membrane in to complete up the picture here, in red as usual. So here's the basement membrane on which these endothelial cells are sat. So in the left heart, because of the high pressures of the blood, it is far more common for endothelial cells to be destroyed and for you to get little gaps in the endothelium like this. Now, of course it will repair itself. You will produce a new endothelial cell that can go here in due time, but acutely what happens is that you get a little blood clot forming on the surface, because now, instead of having the blood just exposed to the apical surface of endothelial cells, the blood is now exposed to the basement membrane here, which contains all sorts of proteins that the blood would never normally be allowed to see. And these proteins are going to induce a blood clot formation. So the blood has a way of clotting where platelets bind to one another and you form a fibrin meshwork that ties all of the platelets together, and that forms a blood clot. And normally this does not happen inside blood vessels because all the blood sees is the apical surface of the endothelial cells, and these are incredibly good at stopping blood from clotting. However, when the blood sees the basement membrane, which is full of things like collagen, those things activate the blood to clot. So now, when you've got this endothelial cell missing from here, you will get a blood clot forming on top of here, a tiny little one, and this is what is known as a vegetation. So, let me just draw this in. So these are, undo that, go back to the thin pen. Okay, so these are platelets here that are all bound together as part of the blood clot. So they're all binding to one another here, like so. And then binding the platelets together and giving the whole thing a lot of strength, there will be a fibrin meshwork. And I'm just deciding what colour I'd like to do the fibrin meshwork in. I think I'll go back to purple. So in between the platelets, you'll have a really dense meshwork. And I can't really even do this justice. You'll have like a spider's web of little protein fibres connecting all of the platelets together and giving this thing a lot of strength. And these are the two components of a blood clot. So a vegetation is a little blood clot that is formed on the surface of the exposed basement membrane, and this has two components. It has the platelet component and also the fibrin meshwork. Now, Again, I'm not going to go through how a blood clot actually forms because, again, it takes me absolutely ages to go through all of the mechanisms as to how this forms, how the platelets get activated to stick together, and how the coagulation cascades are activated, in this case the intrinsic coagulation cascade, to produce the fibrin meshwork. But we'll just state that this has happened. Again, I've got videos, I think in the cardiovascular system playlist, which go over exactly how hemostasis occurs, the formation of a blood clot occurs. But for our purposes, we'll just go from here and accept that it does happen. So, where you've got this exposed basement membrane, then you get this vegetation forming, which is this little blood clot where loads of platelets have stuck together, and you've got the fibrin meshwork here in purple, which is holding it all together and giving it structural integrity. Now, this thing is not so good at stopping bacteria from sticking to it. So these streptococci, these viridin streptococci or these Staphylococcus aureus bacteria, they can stick to this thing. They can't stick to the apical surface of the endothelial cells, but they can stick to this pre-existing vegetation. And this is the reason that infective endocarditis is far more common in the left heart than in the right heart, because you are far more likely to have pre-existing vegetations in the left heart, especially on the left heart valves, than you are to have them on the right heart valves, just because the pressures are so much higher in the left heart. Okay? So let's imagine now that indeed we do have a pre-existing vegetation, and we'll imagine that it is actually on one of the heart valves, okay? So that we're going to get infection of one of the heart valves. Alternatively, it could have been on a portion of the endocardium very close to the heart valve, and then the infection could have spread into the heart valves. But just to make this as basic as possible, let's imagine that we have this pre-existing vegetation on the endothelium covering one of the heart valves, and now what we're going to get is the bacterial cells sticking to this. So let's draw a bacterial cell now, and I'll do this in blue, and I'll have it as a thick pen here. So here are the bacterial cells that I'm just drawing as blue blobs here. They're coming in the blood, and what they can now do is stick to this, and they can go into this vegetation. So here we have some of the cells going into the vegetation, like so. So this is how they can actually begin the infection of the endocardium, by binding to pre-existing vegetations, and these are far more likely to exist on the left heart than the right heart. Right, what's now going to happen is, of course, that sentinel cells underneath the endothelium here are going to acknowledge the presence of these bacteria. These are real pathogens now. They will have all sorts of molecular molecules that are, that no human cell would ever produce, and some of these will end up being secreted off. Indeed, some of these bacterial cells will now even go into the connective tissue underneath. So this, remember, we're imagining is the endothelium over a valve cusp, so underneath we'll have the layers of connective tissue. Some of these bacterial cells might go into those layers of connective tissue underneath, and there they're going to activate sentinel cells, which is how you're going to get the inflammation. So let me draw some of these sentinel cells now. So we'll begin with a resident macrophage in this underlying connective tissue of the heart valve cusp here. So all tissues in the body have sentinel cells, including the heart valve tissue. So here is a resident macrophage, and then you'll also have another type of sentinel cell that's in many different tissues around the body, known as a dendritic cell. Now, we haven't previously discussed dendritic cells in this video, so let me just do a little introduction of them. So they have this star-shaped structure with lots of dendrites coming off their central cell body, like so. Now, they are absolutely nothing to do with nerve cells, which also have dendrites. They are not capable of conducting action potentials. They are named because of the structure that they have these dendrites, not because they have any similarity to neurons. So this cell here is a dendritic cell. And the purpose of these dendrites is so that the dendritic cell can stretch its arms into loads of different areas of the tissue so that it can look for pathogens. So it's a sentinel cell, that's its major job. It looks for pathogens present within the tissue, and if it finds them, it will set off the alarm signal, it will set off the inflammatory response. And this one over here, this is a resident macrophage within this tissue. Okay, so when you get this bacterial infection in the vegetation, as I've discussed, what will happen is some of the bacterial cells now will actually come down into the underlying connective tissue underneath here, and also these cells will be secreting molecules or releasing molecules that will go down into the underlying connective tissue. The point is that these cells will be exposed to molecules that should not be present in that tissue, molecules that no human cell in its right mind would ever make, and these are going to alert these sentinel cells that there is a pathogen present. So the actual presence of this pathogen is going to activate the sentinel cells here, and they are of course going to start releasing those famous pro-inflammatory mediators that we have learnt before, which are interleukin-1 and tumor necrosis factor alpha. So they appear again. So interleukin-1 and tumor necrosis factor alpha, TNF-alpha, and these are now going to cause inflammation in this area. And it's still a little bit controversial as to how you get the inflammation in the area, as to whether the interleukin-1 and TNF-alpha are going to act on the endothelial cells covering the valve, or as to whether there are blood vessels actually in the valve connective tissue that they're going to act on. But what's certainly going to happen is you are going to get inflammation in the heart valve. You will get an exudate forming, and you will get leukocytes, white blood cells, being brought in here. So, in particular, the white blood cells that you'll get brought in will be macrophages, and for short, I will abbreviate macrophages down to M5. This is one of the abbreviations for macrophages, M and then a phi, like so. And you'll also get neutrophils, which are a cell that we haven't discussed previously, and again, there's an abbreviation for neutrophils, which is N, and then the Greek letter phi there as well. So I'll just, again, do a little introduction to neutrophils. So neutrophils are another phagocyte. They're a cheaper type of phagocyte than a macrophage. They're a smaller type, and they have this classic nucleus that is multi-lobed. So they don't have multiple nuclei, but they have a very strange shaped nuclei where it has multiple lobes, classically 3, like so, with little tubes connecting the lobes of the nuclei. Okay? And it's not necessarily just 3 lobes. You can have 4 or 5 lobed nuclei in neutrophils. And as I say, they are a phagocyte, so they engulf pathogens and break them down, just like macrophages. They're not as good at doing it as macrophages. Macrophages are stronger than neutrophils. Okay, so what's going to happen then is in this underlying connective tissue of the valve, you are going to get an exudate forming, and you're going to get white blood cells, particularly these 2 types of phagocytes brought in. Now, this would be fantastic. It would work fantastically if the bacterial cells were all present in this underlying connective tissue. If they were all in here, these would have no trouble clearing out this infection, doing away with this rabble, and sorting this whole thing out. The problem is that a lot of these bacterial cells are up in the vegetation, and I'm afraid to say that these cells of the immune system and also the proteins of the immune system, the complement proteins, find it very, very difficult to attack the bacteria that are in the vegetation. So they will clear them out if any bacteria dare to come into the connective tissue underneath, but they will fail to clear the ones that are actually in the vegetation. And this is why this disease is so dangerous, because the immune system can't really deal with it. It can't get to the ones that are actually in the vegetation. So what's now going to happen? The ones that are in the vegetation will divide and divide and divide, and the immune system won't be able to do anything about it. If they dare to come into the connective tissue underneath, they will be destroyed. But of course, we can't annihilate the actual source of them, which is in the vegetation. So you're now going to get, whoops, didn't mean to do that in yellow, loads of them. Here's another one, here's another one, here's another one. So you're going to get loads of them forming in this vegetation. And they'll continuously be spreading from the vegetation downwards into this connective tissue, and there you'll get the fight between them and the immune system. The immune system will win, it will get rid of them, but it will lead to massive tissue damage as it does. So you'll continuously be having this fight, which will be destroying the tissue of the valve underneath. In addition, up in the actual vegetation here, these are going to start attacking the neighbouring endothelial cells. So these neighbouring endothelial cells are going to start dying. And what that means is that the vegetation is going to therefore expand. So going back to the purple colour, this thing is going to start expanding this way and expanding this way. So the vegetation will grow and grow and grow. You'll get more platelets and more fibrin meshwork laid down, and the bacterial cells will say, "Whippy, more home." So their house will be expanding, their population will be going up and up and up, and the immune system can't attack them. It can't get into the vegetation. So this is extremely dangerous. So what's happening here? The vegetation is expanding. So to paint a full picture then of this, if this is occurring on one of your heart valves, and I will just draw a picture of the mitral valve again, which remember is the major one that is most commonly affected by this. And then after that it's the aortic valve. So here's the mitral valve, here's the anterior cusp, and here are the three scallops of the posterior cusp. So if I draw a vegetation on here, and I regret now drawing the thing in purple because I'd have liked to draw the vegetation in purple, I'll go for a different colour now to show the vegetation, and let's go for a darker purple, a more bluey purple. So let's say we've got this vegetation then here. It would have initially started off as a pinprick. So it would have initially started off as something like that pinprick that I put right at the centre there. And now it's expanding because of the bacterial infection. And it's now this entire bit here, and it will just keep expanding, and underneath that vegetation, the connective tissue underneath there is being destroyed because of the continuous battle between the cells that are moving down and the immune system, and that will result in huge tissue damage. Remember, these cells use horrible tactics to clear these things. They'll be releasing all sorts of foul molecules, like the reactive oxygen species that we talked about when we were discussing calcification. So you'll be getting massive tissue damage here. And the vegetation will just keep expanding. So it can expand all the way over here. It will expand and expand and expand, getting bigger and bigger. And then you can imagine that it will start destroying the edges as it gets to the edges. It will destroy them, and you can imagine that the cusps will now start to shrink down. You can also understand how this infection can lead to holes forming and how it can lead to caudal detachment. So the vegetation will just expand and it will be destroying all of the tissue around it. Right, and this cannot just occur to the endothelium over the heart valve cusps. We're particularly interested in it occurring in the endothelium over the heart valve cusp, but it could also be occurring on the endothelium over the actual wall of the heart. Now, coming back to the two different bacterial species that can actually cause this, Staphylococcus aureus and Viridans streptococci. I'm sorry about saying two different species. Viridans streptococci contains many different species, so it's more than two different species. If it is Staphylococcus aureus that is actually causing this, those vegetations will be expanding at a very, very fast rate, and therefore it's very, very dangerous. The disease progresses very, very quickly. Destruction occurs very, very quickly. Whereas if you're slightly luckier and it's Viridans streptococci that is causing this, then it will progress at a slower rate. The vegetation will expand at a slower rate. The disease will progress at a slower rate. Hence why we divide this into acute and subacute, meaning slower than the acute form, infective endocarditis. Okay, so let's now go on to discuss how this can actually kill you. So if this is not treated, and it does need to be treated, the immune system will not deal with this now. It will kill you if it's not treated. And of course, the treatment is either antibiotics, and if antibiotics don't work, then you may even need surgery to remove the infected heart valve and replace it with a prosthetic one. But hopefully antibiotics will work to clear the bacteria. If you don't get treatment for it, however, how does this actually kill you? Well, there's two ways that it can actually kill you. So, two causes of death. One of them is that it can send off septic emboli, which can go up to the brain and cause a stroke. So, remember that the vegetation that's expanding, it's made up of blood clots. It's made up of platelets bound together by fibrin mesh. If a part of that detaches from the endothelium, so imagine that this now detached from the basement membrane, rather, and flew off in the blood. Where's it going to go? Well, we're imagining that this is the mitral valve, that this is happening. It's going to go into the left ventricle. The left ventricle is going to push it up into the aorta, and it can then go zooming off up one of the carotid arteries to the brain, and then it can block some artery in the brain. It's a septic embolus because it's infected. Septic refers to the fact that it's infected. It's full of bacterial cells. Emboli is just something, a particle floating around in the blood. It can go off up to the brain, and it can block a blood vessel in the brain, and that can lead to a stroke. So that's one of the ways that can lead to death. And of course, stroke is just cerebral ischemia, a huge part of the brain dying because of lack of blood supply. Okay, so that's one way it can lead to death. The other way is that it might lead to heart failure that progresses extremely quickly. Now, usually heart failure is a slow process where the heart gets gradually weaker and weaker. If this destruction of the heart valve is so bad so quickly, imagine the whole thing gets destroyed, then it might be that the heart completely fails to function anymore. The left ventricle might, instead of pumping blood into the aorta anymore, just pump it all back into the left atrium, and then it goes back into the pulmonary circulation. You might get complete failure of the heart to actually perform its role at all. And if that occurs to a massive extent, it will kill you because no blood is going around your body, and therefore your brain will die because of that. So those are the two major ways that infective endocarditis can kill you. So it does need to be treated by antibiotics. It is a medical emergency. Okay, now even if it is treated by antibiotics, so I'll go down here. So if it is treated by antibiotics, in the long term, you can still be left with a huge amount of valvular damage. So antibiotics might save your life. It might stop you getting a septic embolus causing a stroke or destruction of the heart valve completely and therefore heart failure due to that. But you still might be left with the cusps being smaller than they should be. So I'll draw this out again. So imagine the cusps are now left like this after this episode of infective endocarditis. You might still be left with lots of the chordae tendineae no longer attached, and also maybe holes in the cusps. And that's now going to lead to regurgitation of that heart valve. Now, the regurgitation might not be bad enough that the heart completely can't function and therefore it kills you instantly, but it will chronically lead to valvular heart disease, and then chronically lead to the vicious cycle of heart failure where the hypertrophy occurs and you gradually get weaker and weaker and weaker. So, even if you're successfully treated with antibiotics, it can still lead to permanent damage and therefore valvular heart disease. And the two major heart valves that are affected, I'll remind you as we finish now, are the mitral valve and also the aortic valve. Okay, so we will finish our discussion of infective endocarditis there then. We'll have a break now. In the next video, we'll go on to myxomatosis degeneration, and in particular, we'll discuss the disease Marfan syndrome.